Good afternoon, ladies and gentlemen. It's my honor to welcome you to our webinar, Shimadzu Solutions for Honey Testing. This webinar is the perfect place for you to understand the various techniques for honey testing and an opportunity to talk to our experts and have your queries answered. So whether you are a beginner or an expert who needs a refresher on chromatography or mass spectrometry techniques, so this webinar is a perfect place to begin. So over the next few minutes, you will be taken through a complete solutions from sample preparations to accurate analysis, the various applications for honey testings and the method packages that are available to make honey testing fast, accurate, and simple. This webinar is brought to you by Spinco Biotech, India's leading analytical instrumentation company, which was started in 1981. True to its tagline, Spinco is bringing technology and enabling science for the past 41 years. With over 800 Spincoids and 50 offices across India, we are closer than ever. In the recent past, we have had 100 application specialists, and we are targeting 150 application support for your various application needs. So it gives me immense pleasure to welcome our panelists for today, Dr. Sagar Utture, Manager, Customer Support Center, Spinco Biotech Chennai, who later today will be launching our application support for honey testing booklet. Dr. Madhusamy, LCMS Specialist and Manager, Mr. Abhishek Shembekar, Business Development Manager, and Mr. Devajit Sen, Territory Manager. I would like to welcome Dr. Madhusamy, our first speaker, and his title is Honey Quality and Safety Testing by LC and LCMS-MS. Dr. Madhusamy has 12 years of experience in food testing and a significant knowledge of ISO 17025, EIC, APEDA, and food safety standards. He has extensively handled instruments like LCMS-MS, GCMS-MS, HPLC, GC, FTIR, ICP-MS, and AS. Prior to this, he has obtained his PhD from Upasi Tea Research Institute under Bharatiyar University, and he has done his master's in chemistry from Anja College under Madurai Kamaraj University. He has worked with reputed companies like Bureau Veritas Chennai, TV Sud Bangalore, and currently working in Spinco Biotech as an LCMS specialist. Please welcome Dr. MS for his first presentation of today. So before we start, a few housekeeping rules. Please type in your queries in the Q&A box only so that we can answer your questions at the end of this session. Over to you, Dr. Madhusamy.
Yeah, thank you so much for nice introduction, sir. Let me share my screen.
So before Dr. Madhusamy gives his presentation, I'll have a small poll question. So that just to understand the customer's requirement. So the first question will be, so hope you can see it on the screen. The trace levels of antibiotic analysis in honey is qualified by using dash. So the options are HPLC, LCMS-MS, GCMS, and ICP-MS. So you have these four options. You can choose one of these four options. So I repeat the question once again. The trace level of antibiotic analysis in honey is quantified by using dash. Is it HPLC, LCMS-MS, GCMS, and ICP-MS? Thank you all for participating. I'll just end this poll now. So the results are Dr. Madhusamy, 98% believe that LCMS-MS is the technique for trace level and trace level antibiotic analysis in honey, and 2% believe HPLC is the. So over to you.
Yeah, thank you, sir. Very good afternoon, all. Really, I'm happy about very well known about the honey analysis in LCMS-MS. I'm happy about your answer. So today I'm going to present about honey quality and safety testing by LC and LCMS-MS. In this presentation, I would like to discuss about current status of honey industries and its regulation, LCMS-MS for contamination analysis workflow, application for contaminant analysis in honey, and followed by I would like to highlight about honey quality identification through sugar profile analysis and data processing workflow. Let me discuss about current status of honey industry and its regulation. First, we need to understand what is honey. Everyone knows very well about honey is a natural sweet substance produced from the nectar of the plant flowers by honeybees. As per the research, Indian apiculture market may reach a value 33,128 million by 2024. It clearly indicates that to the testing laboratory, honey manufacturing and honey supply in the market will increase in future all over the world. The growth of honey based on the ingredients and food manufacturing. For key ingredient, they are using in the Ayurveda as well as the pharmaceutical, because we know honey has the nutritional high content of the nutritional value as well as the medicinal properties. For food manufacturing, it was used in the bakery products manufacturing or confectionery products, jams, etc. There are many types of honey available in the market. Based on the origin, based on the plant sucking insect, it was divided into multiple types. Based on the origin, there are two types. One is blossom honey or nectar honey dew, and based on the plant sucking insect, there are 6 types of honey available in the market. In recent days, when we look at the newspaper, we may identify many highlighted news about the honey. For example, neonicotinoids insecticides are found in 75% of global honey samples and half contains a cocktail of chemicals. Mostly they are targeted contaminants is neonicotinoids. Also, we can see most honey brands fails advanced quality control testing when it was supplied to local market as well as the export. And we may found many kind of antibiotics with a different level identified from the well-known brand of honey sold in the market. How it was entered into the honey? There are 2 ways to enter this kind of antibiotics. One is treatment, another one is practice. Treatment means there are many kind of the antibiotics used for bacterial disease like chloromenical, tetracycline, erythromycin, lingomycin, streptomycin, etc. These are the antibiotics used for bacterial disease, as well as the antibiotics residues increase based on the improper beekeeping practices. There are different regulations available for honey local market supply as well as the export. For domestic, we must have follow FSSAI, Food Safety and Standard Authority of India, and AGMAR, Bureau of Indian Standard. When we are going to export from honey from India to other countries, we have to follow EAC as well as international guidelines like Codex and EU regulations. For regulation in India, we have to follow Food Safety and Standard Authority of India. They have list out the antibiotics list, and the list was fixed by the tolerance limit. These are the tolerance limits are established basis of the LOQ identified when analyzed using LCMSMS technique. For export, EU regulations and codex, they never mentioned any tolerance limit or ML value. It clearly indicates the antibiotic should not allowed to use in the honey manufacturing process. These are the current status. We have to understand why LCMSMS for this kind of contaminant analysis. There are many factors. First one, higher sensitivity. We know very well, regulations always updating their MRL value is very, very low. For this case, demanding this MRL value as per the LOQ value, we have to use higher sensitivity instrument like LCMSMS. Also, LCMS is called a universal ion detector because it will determine the ions which comes from the molecules. The LCMS having high throughput because nowadays the LCMS supply along with the method package and library, which was used to analyze many compounds or which is used to develop a method quickly without any struggle. Also, the mass spectrometry is used for qualitative and quantitative analysis. The mass spectrometry can be used to analyze multiple compounds in a single run. So the speed and diversity of its mass spectrometry is very high. Also, we can achieve very lower detection limit when we are using mass spec LC-MSMS. Why we have to prefer go with LC-MSMS for antibiotics analysis? Because antibiotics only we can do it in the LC-MSMS, not in the GC-MSMS. That is the first thing. Second one, the antibiotic analysis, as per the regulation, we need 4 identification points. What is identification points? Based on the mRNA transition. The mRNA transition is an important role in the triple code, and it will differentiate the triple code from the single code. So 4 identification points based on the precursor ion and product ion. For here, unauthorized substance, currently EU 808 clearly mentioned that RPA value for some compounds. These are the RPA compounds. We need to identify 5 identification points. The 5 identification points, 1 for precursor ion and 1.5 for products and 1 for RT identification, separation. So these are the regulation clearly mentioned why we have to use LC-MSMS for antibiotics analysis. This is the 4 typical analysis workflow for honey testing. First one, sample preparation. We know very well when we are working at the laboratory, 5 metric ton of food sample or honey sample, we have to decide it with 5 gram of the sample or 0.5 gram of the sample. So 5 gram of the sample decided multiple tons of honey which was exported from India to other countries or local supply to our Indian market. So next sample preparation, we have to think about sample extraction and cleanup process. This should be more simple process, also convenient for laboratory complaints. Third one, instrument analysis. Fourth one, data processing. All these process, sample extraction and sample preparation almost cover 70%, 20% covered by data processing, only 10% covered by the instrument analysis. When it comes to method development for honey analysis, we have to highlight 7 points. These are the 7 points is very important for method development. First one, scope of the method. When we are going to identify the scope, we have to identify the target matrix and specification sensitivity based on the instrument available in our laboratory. Also, sample preparations, we have to optimize based on the homogenization, based on the particle size. And sample extraction is a main part of the honey analysis because we have to identify and adjust the pH of the particular solutions or which was used the right organic solvent. Fourth steps is cleanup. When we are doing cleanup, we may use simple dilution process or we may use different material, dispersive solid base extraction material to clean up the solvent. Fifth step is instrumental analysis. When we comes to instrument analysis, there are main points is selection of the column and selection of the mobile phase, selection of the interface. There are 2 types of interface used in the LC-MSMS. One is electrospray ionization, another one is atmospheric pressure chemical ionization. These are the parameters we have to identify right way to do the method development. Finally, we have to use proper software to data processing, whether we have to check linearity, retention time, ion ratio, and accuracy. Everything we have to use the proper software to increase the productivity in our laboratory. After this method development, we have to conclude or identify this method is suitable for our regular analysis. That is called validation. So we have to validate this method to ensure our method is more suitable for this process as well as different kind of the honey which was analyzed in our laboratory. These are the 7 steps when you are going to develop the method. When it comes to sample extraction, there are 2 steps. One is liquid-liquid extraction. Second one, dispersive solid base extraction. When it comes to liquid extraction, it's a very simple 5 steps. One is we have to take the samples, then we have to add solvent, then we have to add chemical which is was extraction and purification. Finally, we will centrifuge. From this centrifuge, we have to take it for removal of sugars or removal of fat or removal of colors by using different material like primary, secondary amine, carbon 18 or graphite carbon black. So this is another 5 steps to simple cleanup steps which was used in the Kochas extraction process. When it comes to the instrument, we have to understand LC-MS, how it works. Because LC in the liquid state, MS in the high vacuum environmental conditions. For these two cases, we are connecting through interface. Interface is the core technology of mass spectrometry. Also, it will evaporate the solvent and ionize the molecules. This is the architecture of the mass spectrometry. There are 3 main parts in this mass spectrometry. One is source, another one is analyzer, third one is detector. This is the 3 main source in the LC-MS. The ionization source is to ionize the molecule as well as the evaporate the solvent. Analyzer is nothing but is a quarter pole. It will allow only the sorting ions based on the software. What is quarter pole? 4 metallical rod kept under equidistance from the center of the axis with alternating polarity switching. So it will only allow the ions based on RF by DC voltage. Finally, the ions are reached to detector. The detector will generate the chromatogram based on the incident ions. When it comes to application for contaminant analysis in honey, I have to discuss about few case studies related to pesticides and antibiotics residue. The case study 1, we have to discuss about multi-residue antibiotics using LC-MS. For this LC-MS, we have to use 12 antibiotics listed in the as per the FSSAI in honey. This method was created with Shimadzu method package because when we are going to optimize the MRM transition for particular compound, we have to spend more time. For in this case, we have to directly take the MRM transition from the method package and we will run the standard. Just to verify the RT is coming or not. We can identify through this method package so that we can call this high throughput observed through mass spectrometry. Also, the sample extraction process is very simple. We know very well honey is high sugar content profile. Also, in this case, we have to dilute this honey and inject in the instrument. When we are going with the dilution extraction process, we have to use high sensitivity instrument for your analysis. In this case, we are done 12 antibiotics analysis. We achieved very good recovery for 2.5 ppb, 5 ppb, and 10 ppb. If anything is less than 70 or greater than 120, also it will fulfill the regulations within RST 20%. This is the linearity graph for antibiotics. The linearity range plotted from 0.1 to 2 ppb. Also, the LOQ value is fixed as 2.5 ppb and dilution factor is 10. Almost it will come around 0.25 ppb in the linearity. Second case study related to streptomycin. Streptomycin is the aminoglycosides mainly used for treat honey hives. Also, this streptomycin we cannot analyze directly in the mass spectrometry. We need special ion pairing region to analyze this streptomycin in honey. This streptomycin linearity was plotted from 0.5 to 20 ppb. We have spiked with a different concentration 2 ppb and 5 ppb. We have achieved the recovery is 84% and 104% for 5 ppb spiked level. The streptomycin analysis is done with this extraction procedure: 5 gram of sample, 20 ml of extraction buffer, followed by vertex, and we have filtered through Wattman filter paper. Finally, we adjust with pH 6.5 plus or minus 0.5. After adjustment of this solution, we pass through the HLB cartridge. Once passed, finally we elute with combination of formic acid, isopropanol, water with a different ratio along with 0.15% of the HFPA. The linearity of is this streptomycin, we can achieve greater than 0.99. Also, the RST is less than 13.5%. So this kind of streptomycin is very, very useful when we are analyzing the honey. So complete solutions we have developed for this streptomycin analysis in honey by using LCMSMS. Case study 3 related to pesticide residue using LCMSMS. When we know very well neonicotinoids, we are focusing pesticide residue in whether it's a LCMSMS or any other technique. Why we have to choose LCMSMS? We have to achieve very low level concentration and we can ensure our honey is very good level for safety, for human being safety. Also, this kind of pesticide residue analysis, we can do it with a different spiked concentration and we spiked almost 244 pesticides. In these 244 pesticides, 190 pesticides achieved within the range. It means 70 to 120%. And remaining pesticides, almost 7 pesticides less than 70% recovery and 37 pesticides greater than 120% recovery. For this kind of pesticide residue, we are using modified Koch's method. It's nothing but we are taking 5 gram sample, we can add ethyl acetate. Finally, we'll vortex, centrifuge, and we may dilute with, it means we have clean up with 50 mg of primary secondary amine, and finally we will evaporate it by using nitrogen gas. And we may dilute for methanol along with formic acid for LCMSMS analysis. In this case, we have developed a very good method to achieve very low level of 5 ppb level. In this LCMSMS analysis, the linearity was plotted from 0.2 to 20 ppb. These are the fipronil, fibrimate, and pyridabene. We are using it's a challenging compound to achieve very low level. When you are using high sensitivity instrument, you can easily identify and you can easily achieve the lowest LOQ value. When it comes to pesticide residue analysis, LCMSMS is not only the enough technique for complete pesticide residue test in honey matrix. Also, we have to use GCMSMS for volatile pesticides like organochlorine, organophosphorus pesticide. In this case, we have developed a very good method for pesticide residue, including 214 compounds in GCMSMS. In this, we have spiked 4 different levels. In this, 74% of the 74% of recovery achieved for LOQ in 5 pp level. Also, the linearity was plotted 0 to 50 ppb. The recovery of 207 compounds achieved 70 to 120, less than 7 compounds achieved less than 70% recovery. For this pesticide residue analysis also, we are using modified cultures. Just we will dilute with ethyl acetate and inject in the instrument. When it comes to honey quality identification, the sugar profile analysis is very, very important. For this case, we have to use HPLC RID detector. For honey quality ensuring through glucose fructose ratio. The glucose fructose ratio should be 0.4 to 1.6%. For this case, we are using Shimadzu I series along with RID detector and mobile is water ACN 2080. So we can plot out the linearity and achieve the R square from greater than 0.99. Another one important modern technique used in honey is ELST detector, evaporator light scattering detector. For ensuring quality of the honey based on their hydroxymethyl furfural minor compounds presence in the honey. If the hydroxymethyl furfural will increase, the honey quality will affect. As per the codex, the hydroxymethyl furfural value is 40 ppm. This can be analyzed in GC, but the problem is the fructose, glucose, carbohydrate, it's not volatile compound. We cannot analyze in the GC. That is the reason we are using HPLC RID detector. For come to this place, we are using dual detector. Why we are using dual detector? Hydroxymethyl furfural detected only the UV, not in RID or ELST. So we are using UV to identify hydroxymethyl perfural, followed by we are further this UV end is connected to the ELST detector. So dual detector will ensure the quality of honey through HPLC technique. This method optimized within 8 minutes to identify hydroxymethyl perfural. Apart from this case study, Shimadzu having the ready-to-use method package. There are different method package used to develop the instrument method quickly. The benefits of method package is it will include pre-registered MRM parameter with optimized quantitative and reference ion. Also, it will include LC separation parameter, retention time, peak identification parameter for each compound, report templates for outputting quantitation results. Also, we need to discuss about data processing workflow, how it will happen. For example, data processing workflow based on the integration, quantification, QAQC, and reporting. The integration is very important when it comes to the data processing workflow, because we know the common integration error, it may be like a picture of A or B or C. We need to get proper integration base to base. In this case, we have to introduce one function called peak intelligence. The peak intelligence, when we are using to data processing, it will reduce the data processing time and it will increase the productivity of your laboratory. How it will work? For example, just you will choose the algorithm which was available in the Lab Solution Insight software. It will automatically integrate base to base. The peak intelligence is nothing but it will behave based on the artificial intelligence. This peak intelligence will work equal to more than 10 years experience chemist or scientist. So whoever doing the analysis or data processing without experience, they can use this function to easily integrate the peak. For example, peak strength is different. Two peaks eluded nearby, it will integrate automatically the particular peak. We cannot do any adjustment. Also, low signal to noise ratio peak. It means low response of the compounds. It will integrate automatically. Another one, high baseline. If the baseline is very high and peaks also lower the response, then it will identify and integrate automatically with base to base. This kind of the integration will increase the productivity of the laboratory. We have Lab Solution Insight. This is a difficult view of the Lab Solution Insight. In this Insight software, you can see the compound list, sample list, as well as the linearity and chromatogram view. All these views in a single window you can monitor. Also, you can move first compound from 200 compound or 500 compounds. It will easy to move so fast so that you can integrate fast and you can process the data very quickly. Not only this process, always it has the different kind of ion ratio setup based on the regulations. For Sandy, you may use 30% or EU808, you may use 40%. Not only this regulation, if you customer expect any different kind of flexible ion ratio setup also available in our Lab Solution Insight software. Another important source, when we are going to process the data, we have to identify any variations during the analysis. This kind of the variation we can identify through flagging setup. This flagging setup easily highlight your value is very low as fixed by you. It will highlight it in the blue color. If the value is high compared than your fixed value, it will show in the red color. This kind of the review we can do it and we can improve our data processing always as well as the reporting time. Apart from the analysis workflow, the honey testing, we are using different column, different Shimadzu column. There are different type of column used for pesticide and antibiotics analysis. For example, Simpac G series, Simpac Scepter, Simpac Arata. These are the different kind of columns, analytical columns available to test the honey samples in LCMSMS as well as the HPLC. From this presentation, today I discussed the current status of honey and its regulation, regulation for different honey testings, LCMS for contaminant analysis, analysis of multi-residue antibiotics and pesticides by using LCMSMS, which is very, very important for upcoming honey development process. Also, I discussed about analysis of multi-residue pesticides by using GCMSMS. And finally, I discussed about sugar profile analysis for quality of honey and method package which was used for instrument method development in our LCMSMS as well as the HPLC. The another important function called peak intelligence, it will integrate automatically whether the chemist is more experienced or not. It will integrate and it will it will support for smooth functioning of the data processing. Also, we have very good lab solution insight software for easy data processing to reduce the data processing time to report the results. And there are different type of analytical column used for honey testing in HPLC as well as the LCMSMS. These are the total solutions available nowadays when we are going to analyze honey in different analytical technique called LC or LCMSMS. Thank you so much for your valuable time.
Yeah. Thank you, Dr. MS, for giving us this complete workflow required for honey analysis, starting from a sample cleanup to validation, analysis, and data processing. Of course, I'm sure a lot of people will have a lot of queries, so we'll get back to you at the end of this session so that you'll be able to answer their queries. So our next speaker is Mr. Abhishek Shambekar. So he's currently the business development manager in Spinco for the past 13 years. So he started his career in Spinco as a product specialist, and now is the business development manager working for Spinco in the past 13 years. So he's a business manager who always puts the customers first. His thorough understanding of the subjects makes him a technical solutions provider. So he will be presenting about Shimadzu UFMS for honey analysis. Over to you, Abhishek.
Yes. Hello, everyone. I am Abhishek Shambekar from Spinco Biotech. Today, I will give presentation on Shimadzu LCMS and its utilization towards food how it is useful for the contaminant analysis in honey. LCMS is a very useful and a powerful tool today for any qualitative and quantitative analysis. The ever-changing demands of each industry require not only the right people, but also the best technology. Triple quadrupole mass spectrometer is a versatile tool that can be adapted to a wide range of applications. In terms of instrumentations, researchers require speed, sensitivity, and the flexibility to adapt to meet the new challenges. For years, Shimadzu has been known as the leader in liquid chromatography, and now Shimadzu is being recognized as a leading manufacturer of LCMS triple quadrupole mass spectrometers. Shimadzu offers world's fastest LCMS instrumentation, all without compromising the sensitivity. No longer settle for anything less than the best. So with this, I would just like to start with my presentation. I believe all of you would be aware about Shimadzu, but just to again mention, Shimadzu Corporation Japan is the longest serving analytical instrument manufacturer in the world. Since 1875, Shimadzu has been introducing various cutting-edge technologies and serving the scientific community with their innovations. Spinco Biotech, which is the channel partner to Shimadzu, 41 years we have been there into the industry by bringing technology, enabling science, and headquartered at Chennai. We represent Shimadzu for their entire liquid chromatography and mass spectrometry product line. We have 33 offices across the country with a team of more than 700 people to support all our customers. We also have a state-of-art lab, our customer support center at Chennai, to support all our users in terms of training, method development, or any other kind of support. Since inception, customization has been a cornerstone of our company culture. Shimadzu is a one-stop solution for all your analytical instrument needs, be it a UV spectrophotometer or an high-end LC-MS/MS system. All the instruments are manufactured by Shimadzu. So it is a true one-stop solution where you can you have a UV spectrophotometer or an HPLC, LC-MS/MS system. So all the instruments are manufactured by Shimadzu. Not just that, but Shimadzu is also blessed to work with Dr. Koichi Tanaka, who has got a Nobel Prize for his work on MALDI in 2002. Even in terms of patents, when we talk about innovations, innovation has been the DNA of Shimadzu. Shimadzu has got the largest number of patents in the industry in comparison to any other vendor. So coming on to the subject today, LCMS, when we talk about mass spectrometry as a technology, Shimadzu last year celebrated 50 years of MS innovation. It is Shimadzu last year completed 50 years. It was 1970 when Shimadzu introduced their first magnetic sector-based mass spectrometer. Those days it was called as LKB 9000 instrument. And over these years, Shimadzu has been introducing various cutting-edge technologies, be it a GCMS or an LCMS, multi-top technology, and the latest and the very differentiating UFMS, ultra-fast mass spectrometry. I'll be talking about the UFMS technology, what is the significance and benefit of this technology in my coming slides. So before going on the products, I would just like to share with you, MS today is being used by all the different kinds of industry, be it pharma, be it agrochemical industry, clinical and diagnostic, herbal industry, and all the other industry, and very specifically by the food industry towards analysis of various food contaminant analysis, whether it is pesticides, antibiotics, aflatoxin, and various other contaminants. So these are the few references, and specifically from the food segment, like the global and local testing labs, few of the key labs who are involved into the honey testing facility are TUV, Eurofins, Vimta Labs, Intertech. So these are the and more. These are the few testing labs. They have been doing complete honey analysis and have been using multiple Shimadzu mass spectrometers across different locations. Not just the testing labs, but even in the recent times, we have seen the dairy industry, the dairy federations, whether it is a dairy cooperatives or even private dairy like the Hudson Agro, they have been also involved in honey testing, other than the milk and milk product analysis. So recently, we have also seen Amul has come up with a brand of honey. They have started manufacturing honey, and various other dairy cooperatives are coming up with a brand of honey. Other than that, all the food manufacturers, food processors like Patanjali and the other food processors, they are also involved in improving the internal quality as well as to meet the domestic FSSAI or the export regulations. So these are few of our customers who have been using Shimadzu LCMS for different kinds of analysis. So before going on to the product and the features and the benefits, few specific challenges which as a customer, as a user, you would be facing and would be expecting from LCMS. The regulatory agencies, they demand lower reporting limits. We have been seeing year on year, there have been revisions, whether it is an FSSAI, MRLs or EU regulation. There have been several revisions in terms of the reporting limits and the growing contaminant list, whether it is pesticides or antibiotics, the list goes on, is increasing year on year. So from the food manufacturers, suppliers or distributors, the lab basically, they need faster analysis, faster results and accurate results. Laboratories would like to deliver results faster. In terms of commercial testing labs, the competition is very high. They would like to do the complete analysis, being competitive, and overall to control the operating cost of the instrument. So how Shimadzu LCMS system is able to meet these requirements is we have this differentiating technology called as UFMS technology, which is called as ultra fast mass spectrometry. UFMS has been a trademark of Shimadzu, and this technology revolves around three main specifications or parameters: speed, polarity switching, and sensitivity. So ultra fast scanning speed, ultra fast polarity switching, ultra fast and ultra high sensitivity. These have been the pillars of this differentiating UFMS technology. When we talk about different models which are available, so we have range of models available from LCMS 8040, LCMS 8045, LCMS 8050, LCMS 8060, and the very latest LCMS 8060 NX, 8060 NX. So NX 8060 NX has been the top of the line highest sensitivity model available in the industry. So the entire range is available. So in terms of all the models differs in terms of sensitivity. So whichever the model suits best for your application. It is not always we should have a highest sensitivity model. It is rather what model or what sensitivity needs or suits your requirement. So that is where you can select the best model for your application, for your need from the available options. So LCMS 8060, this was introduced by Shimadzu last year. This has been the top of the line, best in the industry available model, and it brings a lot of innovative technologies and few of the patents which are available, like the ion focus technology. So there has been a new and improved ion source with this ion focus technology, and the newly engineered ion optics, which improves the ion transmission efficiency, and the dual ionization source. Here, both ESI and FECI analysis can be done simultaneously using this source. 8045, which has been the blockbuster model for Shimadzu. So the key specifications for this model is ultra fast scanning speed of 30,000 Daltons per second, ultra fast polarity switching of 5 milliseconds, and ultra fast MRM 555 channels per second. In terms of sensitivity, for 1 picogram of reserpine, it gives signal to noise of 4:1. The very unique thing about Shimadzu LCMS technology is the on-site upgradation. So this feature is very specific and unique to Shimadzu, and it gives you the future proof investment. So if you have an 8045, 8050, or any model from Shimadzu, these models are scalable. So suppose if you have an 8045, 8045 today is able to meet all the Indian as well as export regulations, and it is able to meet all the near future and future requirements. But still, say after 5 years down the line or 7 years down the line, the requirements in terms of reporting limits have gone down. Still, instead of investing on a new mass spectrometer, you can upgrade the existing MS to a higher sensitivity model. So if you have an 8045, which is the latest and the most suitable model, 8045 can be upgraded to 8060, which means the 3 times improved in sensitivity, or 8050 can be further improved to 8060. So almost 3 times to 9 times the sensitivity can be improved, and all this can be done in your lab itself. Instrument will come with a our engineer will come with a specific kit, upgrade kit, and they will do the replacement of certain parts, and the instrument will be upgraded to a higher sensitivity model in your lab itself. So to complement a fast MS, we talked about fast UFMS as a technology. It comes with a UHPLC, the N series, the latest N series UHPLC, which comes with analytical intelligence as a technology, features of like AI, artificial intelligence, or Internet of Things, machine learning. So these are the buzzword today in all the industry which we talk about. and they have been incorporated into this system, and combining Shimadzu LCMS with this N series analytical intelligence UHPLC offers flexible workflow from instrument preparation to analytical data processing, ensuring high quality data analysis functions like auto startups, column friendly flow pilot function, and mobile phase monitoring. It is a fully automated UHPLC. From startup to shutdown, everything is fully automated. So when we say startup and shutdown, it does not limit only to startup and shutdown of the instrument, but the entire routine activities like auto purging, column equilibration, baseline check, system suitability, everything can be done by system itself. So it brings a lot of unique features and an advanced instrumentation technology to your lab. So it becomes a best and most suitable front-end UHPLC for mass spectrometer. The construction of UFMS technology, Shimadzu UFMS series mass spectrometers are having a linear design to provide the fastest scanning speed in the industry, fastest polarity switching. It has got steps. Earlier, my colleague Dr. Madasamy explained about the different stages of ionization. So it has a heated ESI source. There are two types of ionization sources, ESI and APCI. So as you know, ESI is used for polar compounds and APCI for non-polar mid-polar compounds. The changeover of these two sources, the ionization sources, is very easy. It is plug and play type. So the user doesn't have to struggle to change these sources, and the changeover is very easy without any tool. Then we have this the DL line, ion transfer optics, from where the ions enter into the system, and then Q array and ultra fast lenses. So Q array and ultra fast lenses, again, these have been a patented technology by Shimadzu. With this design, ions are precisely converged and the signal losses are minimized. In addition, the efficiency of ionization process is improved with the use of heated ESI source. Together with the enhanced desolvation line and the ion optics, there is an increase in ion production and transmission, which generates high intensity and focused beams, ion beams, leading to higher sensitivity. Further, this is a Q1, Q2, and Q3, followed by a detector. So Q2, again a collision cell, a patented design, ultra fast sweeper collision cell, patented by Shimadzu. It minimizes the crosstalk when you do the MRM analysis. So the impact of speed and over the method design. So taking one example where this is the total time of analysis, which have been, when we compare it with the conventional LCMS technology, the total time of analysis is distributed between stabilization time, I mean the voltage stabilization, the instrument stabilization, the voltage settings, MRM, parameter optimization, everything, the data collection time, and the polarity switching time. Because when you are working on multi-analytes, whether it is antibiotics or pesticides, different contaminants, so multi-analyte, some will get positively charged, some will get negatively charged. So here, the ionization switches from positive to negative. So it takes certain time. In case of Shimadzu UFMS technology, the stabilization time is very fast. So it has got an auto MRM optimization function. The instrument gets stabilized very fast. The polarity switching time is just 5 milliseconds, which is the best in the industry. So you can do both positive and negative simultaneously. That is where you get more window for data collection. So you get more data points are collected over a peak. So that peak shape is very good. The quantification will be better. The percentage RSDs will be better. And simultaneously you can run multiple events one after another and collect more data. So this is one significance of having a fast scanning speed. This is one practical example where on this peak there are 9 data points which are collected. with a percentage RSD of 8.75%. The same experiment, when we are running at 30,000 Daltons per second, there are 30 data points and the percentage RSD is improved to 2.45%. So this is the benefit of having a fast scanning speed, so that even if you have a low concentration of compound, still instrument will collect sufficient data point so that you can quantify the peak. So that is the benefit of having a fast scanning speed. Second is the fast polarity switching. It takes only 5 milliseconds from switching to positive to negative. Sometimes when you would have referred the old publications or old methods, say some 7 years old or 10-year-old methods, you would have always seen the two runs, one separate positive and separate for negative. So this is because the instrument used to have a very long switching time for the polarity. And if you try to club in those conditions, you will compromise on the sensitivity. But with this UFMS technology and 5 milliseconds of polarity switching, you can do both positive ionization compounds and negative compounds in a single run without any compromise in sensitivity. So just as a proof of these two, a combination of ultra fast scanning speed and polarity switching, this is one experimental data. This is a practical data which have been reproduced in the lab, where 647 pesticides are screened in just 10.5 minutes. So this data was performed on LCMS 8045 model, and 647 pesticides we are able to screen in just 10 minutes. So this shows the power of instrument, the benefit of the fast scanning speed and fast polarity switching. I talked about the patented and very differentiating UF sweeper collision cell technology. So here I would like to compare the collision cell design of a conventional collision cell with ultra fast sweeper collision cell design. So when we talk about a conventional collision cell design, as you know, ions travel in an orbital motion inside a vacuum. So when it comes to Q2 for a collision purpose or a fragmentation purpose, they get fragmented with an external gas, argon gas is used for a collision purpose. So when this fragmentation takes place, there is a loss of momentum of these ions as they get fragmented. So because of these, there is a degradation detection sensitivity, because the transmission is affected, ions are getting fragmented. So ions will not reach, 100% ions will not reach to the detector part. So hence there will be a degradation detection sensitivity, and there will be always a chance of having a crosstalk or a carryover. So when you run multiple analytes one after another, there is always a chance where analyte of one analysis may elude in the subsequent one. So you get a carryover or a crosstalk or a false positive information. In case of Shimadzu UF super collision cell design, there is a pseudo potential which is formed. So it is something like a small entry, large exit, where ion comes in, they get fragmented and sweeps out very fast out of a collision cell. So the benefit is, even if there is a fragmentation and loss of some in the movement of ions, still the transmission efficiency is maintained. So we get an improved sensitivity because of the transmission efficiency and almost zero crosstalk. So the combination or benefit of having a UHPLC with the UFMS is basically, why UHPLC is to have a shorter run times, you get a better sensitivity, the resolution will be very good when you use a UHPLC, lower the operating cost because you are having a shorter run time. So and ultra fast MS, you will run experiments with a fast scanning speed, ultra fast polarity switching. You can run ultra fast MRM events one after another. So all this at a very high sensitivity. So that is where we say Shimadzu N series UHPLC plus the UFMS from Shimadzu provides best of the data. So it provides ultra high sensitivity, better specificity, and a higher throughput, which is the basic requirement for any analytical laboratory today. So this is all from a hardware point of view, the features, benefits, significance of the features, everything. But ultimately, the user sits in the front of computer. He will be using software. There is a lot of data which he has to review, he has to interpret. So the software workflow is also very much optimized. It has got a well-defined workflow in terms of whether it is data acquisition, data processing, and everything takes place in a compliance manner. It takes care of all the whether it is a 21 CFR compliance or any other regulatory body. So the lab solutions inside software, which is a very easy to use, user-friendly software. My colleague Dr. Mataswamy explained about this. So it has got a very interactive view, a single window where you get all the information available. You get sample versus compound, compound versus sample, chromatography. So everything in a single window. It has got all the lot of lagging functions. So when you do the data review, so all the outliers, the sample which is getting passed, sample which is getting failed, can be color coded. So it makes the job of user very easy. To support the software and different analytic challenges and different contaminants, Shimadzu has also developed the method packages. These are kind of libraries which comes with the preset analytical conditions. So some of the method packages which are available are residual pesticide method package for mycotoxins, veterinary drugs, antibiotics, and also the GCMS database for pesticides. So just to explain one database, so for example, pesticide residue method package, it comes with 836 pesticides, and it comes with all the information available, all the MRM transitions, LC conditions, so that these are ready to use methods which you have to download on your instrument, optimize it once, and you can start using the instrument. So the method development time is significantly reduced, because as you know, most of the time goes into the development part, where you have to set the analytical conditions, take different trials. So here in this case, with this method packages, you get all the information available. So you have to download it just once, optimize it on your instrument, and start using the method. So pesticide method package, 836 compounds are there. Similarly, there is a method package for mycotoxins, antibiotics available. To support these different challenging application requirements, we also have a team of field application specialists. So these are the field application people. They come from a domain expertise which help you do the complete hand-holding process. We don't only provide an instrument, but we provide a total solution. We take care of the hand-holding process in terms of all the user training, method development support, method transfer support, or any kind of challenges which you face. So we have a team of 40 plus application people, field application people, to support you for all your requirements. So they can come to your lab, they will help you, they'll sit with you, and will train you in terms of your requirement. Additionally, 4 customer support centers equipped with multiple mass spectrometers. So we have a customer support center lab in Delhi. This is a Shimadzu's lab, a complete full-fledged lab, and Shimadzu has an application lab in Mumbai, Andheri, so where they have multiple mass spectrometers, complete range of instruments, and then we have Spinco customer support center at Chennai. So these labs are fully equipped, and with an application specialist team is available, so that you can always visit the lab, you can get training, you can take any kind of support needed. So any kind of method transfer activities, development activities can be done through these customer support centers. Additionally, Spinco, as you know, is known for our services. We set the benchmark in the industry. This is one picture where we have shown we have more than 500 field service engineers to support you in terms of any kind of instrument breakdown or training or any troubleshooting maintenance required. So we are just one phone call away. So this is the largest or biggest service team available with any in the industry. Finally, the cutting edge, which is our monthly journal, our publication. So every month it comes, we come out with a cutting edge magazine, which consists a lot of scientific articles, application notes, different technologies. So every month's issue is focused on different theme. So you can always get subscribed for a copy, whether it is a print edition or a digital edition. At present, more than 12,000 print copies are being circulated across the country. 80,000 plus digital editions are being circulated. We also, I take this opportunity to invite your scientific articles, something which you would like to share with scientific community. You can always share your articles with us, and we can publish in our cutting edge magazine. Please write to us if you would like to have a copy of this magazine. Even in the last month edition of Cutting Edge, there was a very nice article which was published on the Hanukkah honey. Hanukkah, which has been a very rare form of honey. So there was an article on that. So you can please go through that. So just to summarize the entire presentation, LCMS 8045 Shimadzu LCMS has been the instrument of choice for various reasons. One is the multi-analyte methods. You can run all the multi-analyte methods with ease without any compromise in sensitivity. The good chromatographic separation, N series UHPLC, which is the best UHPLC available in the industry, with the analytical intelligence as a technology, use very good chromatographic separation, capable to deal with simple and easy sample preparation methods with support from SpinCo and Shimadzu. We provide end-to-end solution, not only on the instrument, but also on the sample preparation technology. Wide dynamic range, high selectivity, achieve low LOD and LOQ limits, and a very high throughput. Speed, polarity switching, and sensitivity, again, the three pillars for this USMS technology. Thank you.
Thank you, Abhishek, for giving us the various options of Shimadzu's UFMS technology for honey analysis. I think, can you stop sharing the screen?
Yes.
Yeah, so now it's time to launch our much awaited Shimadzu Solutions for Honey Analysis booklet. So I would request Dr. Sagar to do the honors. And to all the viewers here, if you want to get a copy of this by courier, please type Shimadzu in the chat box. So as and when you type in the chat, type Shimadzu in the chat box, we will get it, and then we'll ensure that a copy of it is couriered to your address. Just to give a brief introduction about Dr. Sagar, Dr. Sagar Uttare is currently working as the manager customer support center in SpinCo Biotech Private Limited, and is involved in laboratory management, method development, and application support. So prior to this, he was working in TV Sood South Asia as a deputy manager. Before TV Sood, he has worked at the National Referral Laboratory, NRCG Pune, where he was involved in development and validation of analytical methods using GC and as well as LC-MS. He has a PhD in the field of pesticide residue analysis and having around 15 years of diverse experience in contaminant residue research, method development, and validation, laboratory operations. Currently having published several research articles in peer-reviewed journals and contributed for articles in reputed publishers, is the man behind this application solutions for honey testing booklet. And we'll request him to launch this book. Dr. Sagar, just before that one. Yeah.
Thank you, Satish, for nice introduction.
One second.
Yeah.
So over to you, Dr. Sagar.
Yeah, thank you, Satish, for the nice introduction. So I'll be giving some overview of the booklet. So this is a booklet exclusively prepared for testing of honey. And here we have compiled many applications which are relevant to testing of pesticide residues, antibiotic residues, sugar profile, and elemental analysis. So these applications are developed in Shimadzu lab and customer support center in Chennai in Spinco. So as a chemist, we know that when we start developing a method, we have to refer many sources or literature for getting some insights on method development before actually start working on the method. So the idea of having this booklet was to bring together all the applications which are relevant to honey testing at one place, so that it can be used as quick reference during the method development process. So if you see the contents, we have divided it into 4 sections. The first section belongs to pesticide residue analysis, where there are applications related to pesticide residues analysis using GCMSMS and LCMSMS. The second section includes antibiotic residue analysis, where we have multi-residue veterinary drug analysis using LCMSMS, and then streptomycin analysis using LCMSMS technology. The other 2 sections belongs to determination of sugar profile in honey using HPLC, and the elemental analysis using ICP-MS. So this is the first application where multi-pesticide residue analysis method using GCMSMS is demonstrated, where the extraction of pesticides is done using ethyl acetate, followed by cleanup using primary secondary amine, and the analysis was done using GCMSMS. So here an MRM database was adopted to quickly develop GCMSMS method for 14 pesticide residues. The LOQs achieved here are less than 5 ppb, and the recoveries were well within the acceptance criteria as per SANTI method validation guidelines with very good reproducibility. The second application covers pesticide multi-residue method for analysis of 244 residual pesticides in single method. The sample preparation again followed here is ethyl acetate based extraction and analysis using LCMSMS. So here 244 compounds are analyzed in a very short runtime of 20 minutes, achieving a LOQ well below 5 ppb for most of the compounds. And the method validation results are also meeting the validation guideline criteria, indicating applicability of the method for routine analysis of samples for honey. The next application is on multi-veterinary drug analysis, where simple Quechers-based extraction technique was followed for extraction of 12 veterinary drug residues belonging to various groups such as sulfonamides, tetracyclines, quinolones, and some few more. The usability of co-injection technique of the sample extract along with aqueous phase is demonstrated here for achieving better peak shapes, particularly for the early eluting polar compounds. These compounds were successfully analyzed using LCMSMS to easily meet these regulatory requirements. The next application demonstrates method of analysis for streptomycin, which is considered to be difficult to analyze due to challenges with respect to sample preparation and its chromatographic retention. Here, EDTA-based sample preparation was utilized for achieving optimum recoveries, and the recoveries obtained were within 80 to 120%. LOQ achieved was well within the below regulatory limits with help of high sensitivity of Shimadzu LCMS 8060. The next application elaborates use of HPLC with RI detector for analysis of sugar profile of honey. Here, very good separation of 4 sugars is achieved with a LOQ of 100 ppm, and this method can be easily adopted for determination of sugars in honey with very good sensitivity. The next method is ICP-MS, where ICP-MS is utilized for application of analysis of various metal contaminants in honey. And the ICP-MS technique ICP-MS technique was used to determine mercury in its methyl mercury form to meet regulatory requirements as per FSSAI. So in conclusion, we have released a booklet compiling the most challenging applications or most critical applications relevant to honey analysis. And we hope that this booklet will be useful tool for the scientists working in various institutes and laboratories for development of methods, for improvement of existing methods, and for various contaminants in honey. Thank you.
Thank you, Dr. Sagar, for launching the booklet and explaining about the contents of the application booklet. So we have received a huge response for the application booklet, and we shall receive, and you shall receive the same by courier in the next few days. So our next speaker is Mr. Devijit Sen, who has over 16 years of experience and is currently working with Spinco Biotech for the past 14.5 years. He is the territory manager for the eastern part of India and has a passion for chromatography system, especially GCMS and LCMS. He has a good understanding of GCMS for the past 14 years. Needless to say, he is a self-made expert on mass spectrometry. So he will be presenting about the residual pesticides analysis in honey by GCMS. Over to you, Devijit.
Thank you, Mr. Thank you for giving this opportunity. It's really encouraging to see 100+ customers are still willing to listen further on this very interesting of honey analysis. Thank you to all of us. We would not have come this far your support. I would take you to the GCMS application, GCMS instrumentation basically, honey analysis, the recent developments, the technologies, and all the when it comes to GCMS, basically we'll be covering in the next few slides an overview, the workflow of residual pesticide analysis, the instrumentation part of GCMS, and some application data on honey analysis. As my previous colleagues, Dr. Madaswamy and Mr. Abhishek have already explained you, the most application lies in the LCMS category. But still, if you look at the overall analysis requirement of GC or LC based, we can see that 60% pesticide done on LC based technologies, that is LCMS. 30% can be done by both techniques, where GCMS and LCMS both can be utilized, and 10% dedicated to GC or GCMS based. So this is about how it is regarding the analysis of pesticide. Under RA and under on presence of residual pharmaceutical activities, active pharmaceutical analyte in honey. This has been registered as a serious concern. Coming to the workflow regulation, workflow of honey analysis through GCMS, if we look at the regulations which are in place, EU, Codex, EIC, EPIDA, FSSAI. Basically, India follows the FSSAI. And sample preparation is queutures based and modified queutures. When it comes to the instrumentation technique, GCMS model, what we suggest is TQ8040NX and LCMS 8060NX, 8060NX. Data processing is done through our lab solutions inside software platform. As said, FSSAI is the guideline for honey analysis, which is being followed. Sample preparation, Dr. Madaswamy has already covered, but if you look at the honey sample analysis, you can add water and vortex, and then ethyl acetate, and then vortex, sodium sulfate, vortex, and centrifuge, followed by, it can be divided into two parts. Looking at volatility, it can be GC or LC technologies. This is how it goes. Add NGL support plus PSA, vortex, and then centrifuge. Then you can inject 1 microliter to GCMS. If it is LC, then evaporate and then reconcile with mobile phase vortex and then filter. 10 microliter you have to inject for LCMS technology. Now coming to the question, basic question, why MS? There are few answers to it, because honey is a complex matrix. Retention time is not sufficient for separation. specificity and sensitivity, and we get similar response to almost all pesticides. That's also a concern. It can be coupled with both and MS. What we get is a different management mode. In GCMS, it is EI and technique. This is the picture will be that is guiding kind of MS to get a very quantify the piece much closely as that will. That is why GCMS technique is preferred over single purchases. Shimadzu coupled with OC is the auto injector. Above the tower part is the auto injector part through which you can inject automatically through a robotics platform. It gives smart performance, smart productivity, and smart operation. We'll explain in the few slides. The breakthrough innovation by Shimadzu is GCMS NX series, wherein there are three main steps: startup, analysis, and maintenance. In between, the automatic leak check functioning and automatic tuning judgment, all these steps are automated and with the active time management schedules. That means what is the time for maintenance requirement, the analysis requirement, everything, the time automatically will come. This much time is remaining. This is a general process from startup to shutdown. The general processes you need to follow. You have to first start the 4-line pumps, the rotary pumps. Then you have to start the turbomolecular pump. You have to check the leak, wait for stabilization, tuning, then tuning judgment. All these steps are mandatory for a GCMS operation. Now, what Shimadzu has done, the new breakthrough technology will automate entire process. That is, you have to just start the 4-line pump and then wait. Vacuum level check, TMP, starting off a TMP, again leak check, stabilization, all these parts are automated. And then you have to just do tuning, and then finally you can start your sample analysis with active time management functions. When it comes to smart performance, yes, there are quite a few techniques with Shimadzu GCMS. Heat shield ion source, quadruple with pre-rods, off-axis ion optics, overdrive lens, and UFMS technology. These technologies will be explained in the next slides, which are basically unique technologies which are not available with any other companies. We all know that GCMS technique, ionization source technique is basic technique is 90% goes with EI. That is the hard ionization technique, wherein 70 electron volt is applied, and then molecules are emitted, we get the ions. But in case some 10% cases, it will also be there that you need some molecular information, wherein we used to need to use the CI, the chemical ionization, positive chemical ionization. In that case, when you switch from ionization, one ionization source to another ionization, you would need to break the vacuum. There is a general issue with all the GCMS manufacturers. Like this is explained here. EI analysis, then you have to stop vacuum, then exchange the ion source, then again start up vacuum, then again you can change with PCI. That is the positive chemical ionization source. But here in Shimadzu, we can use a smart EI source. That means source, you need not break the vacuum, and you can do EI and then break the vacuum, directly go into PC analysis, and again you can do EI analysis. It is called smart EI source, which is available with Shimadzu. Another technology wherein there is a shield plate being used from the ion source, wherein the effect of filament temperature is nullified, which will not do any kind of ion burning. That means you are assured that the ion source, ionization source, will reach to the detector. The signal will get. Another technology on the right hand side, and there are some P dots. pre-dots are applied before the main quadruple dots. These pre-dots are acting as pre-filters. That means your main quadruple dots will never get contaminated as such. The pre-filters will act to protect it. And then there is another unique design, which is an off-axis ion optics. If you look at the entire horizontal path, the ion source, quadruple, and then the detector, it is little curvature in design. Some bend is there. This bend or curvature design ensures that all the unwanted material or the neutrals will not enter the detector. So you are assured that even another technology is called overdrive lens, the bottom picture. It is nothing but some optical arrangement with lenses, which will ensure that most all the ions which are passing through the quadruple are collimated and those are directed towards the detector straight away. This will also further enhance the sensitivity. Shimadzu is having 800 transitions thanks to its super technology, which allows this many high transitions to occur. The collision cell makes sure that crosstalk is very, very minimized, which Abhishek has already explained in case of LCMSMS. Similar technology is applied here also. Shimadzu uses ASSP, is another technology called Advanced Scan Sim Protocol, with which at very high scanning speed at 20,000 mass unit per second also, we are not losing any sensitivity. That is, signal to noise ratio is very, very high. Here, if you look at, we'll show that without ASP, ASSP, and with ASSP, what happens. This is the general phenomena, and passes like this through the quadruple. It takes time. With this ASSP on, special protocol, it reaches the detector very, very fast. So chances of crosstalk is minimized, and you get very high amount of signals in the detector, ensuring highest to noise ratio, that is the sensitivity. And very interestingly, at very high scan speed also, you're not losing the sensitivity with this ASSP on. Coming to the maintenance part, you can be very assured that Shimadzu GCMS will make your life peaceful. That means, in other way around, you might have experienced, if you have used GCMS, how painful it may be sometimes or at times, changing the columns or the maintaining the injector port. So here, Shimadzu applies a click tech technology, which is very, very user friendly. Just a small turn, and then you can insert the, you can reinsert injector. This is very much easy. Are also speed liners here from side. Okay, has come and injector port. High sensitivity again, Shimadzu uses C23, which is the latest version, digitized flow controller, advanced flow control. You can see using this, the electronic noise has been just reduced. So you get a very
Looks like there is a technical glitch. We'll just wait for DJ. I think there was a lot of lag, DJ. Is it okay now? Can we continue?
Yeah, I'm sorry, I got disconnected in between.
Yeah.
I could not understand.
Good.
This maintenance part was covered. I could listen to the maintenance part.
Yes. So you can continue from there.
Flow controller. Okay, thank you. Shimadzu uses advanced flow controller 2030, which ensures that the electronic noise is minimized and you get very stabilized state. All the mass spectrometry users are aware of that turbomolecular pump is say that of the more hands get more sensitivity. That means ensures that the right hands so the detector through the quadrupole should be under vacuum, because without vacuum ions will die, it will not reach to the detector. So here the
So seems to be another
KYC,
technique.
meanwhile, I think we can take the questions.
Yeah. DJ, we will. I think there's some broadband issue with your side. So what we'll do is we'll start with the Q&A session, and then we'll come back to you. Probably by that time, your broadband also might be improved. Okay, so we'll start with the Q&A. We have received quite a few question and answers. Before that, I think there's one more poll question. So we'll just have that question. I'll just launch that question. So the query here is, in order to reduce the integration time in the data processing, we use a dash function, peak intelligence, chromatopack, iPeak finder, and all of the above. So DJ, you can stop sharing the screen. So I'll repeat the question once again. In order to reduce integration time in the data processing, we use a dash function, peak intelligence, chromatopack, iPeak finder, and all of the above. So we'll give another 20 seconds for everyone to answer. Okay, 5, 4, 3, 2, 1. So here are the results, Dr. Madaswamy. So 50% of them believe peak intelligence, and 3% believe chromatopack, and 18% believe iPeak finder, and 29% believe all of the above. So your answer, please.
Yeah, definitely peak intelligence give 100% less time compared than the other parameter which was used in lab solution software. Why we have suggested peak intelligence? No, this is a very good function recently launched by Shimadzu. This function will integrate like a 10 years experience, like a scientist. If scientist having the 10 years experience to integrate the peak, that kind of experience and that kind of setups already have with peak intelligence function. So definitely peak intelligence give you a better solution compared than other parameters, which may be the chromatopack or iPeak finder. Thank you, sir.
Okay. So there are quite a few questions which has been posted by the viewers. So I'll just kickstart with one of them. Before that, Dr. Sagar, since you said you are the manager for the customer support center in Chennai, so how does your customer support center support our new user, a person who's actually just purchased a mass spectrometer? So how does your customer support center support a new user?
Yeah, actually at customer support center, we have almost half a dozen mass spectrometers starting from 8030 to 8060 NX, all the models of Shimadzu LCMS. And we have a dedicated staff who are continuously working on method developments for new applications or challenges which customers are facing. So at the same time, we have, as Abhishek had showed in his presentation, that we have field application support team who, if a new user of MS, he doesn't have much experience towards handling of LCMS. So the team do handholding and do the training, on-site training of the LCMS software and hardware part. Along with that, they does help them to do the method transfer and method development part also on their instrument. And at the end of suppose like 2 or 3 months time, we also take the users to the advanced level of training. So this is the basic role of CSC, and through which we try to support customers in the initial phase of MS handling.
So to understand, so does it only suffice to method development, or do you also support for the validation, complete method validation and other things?
We do support for method development, method transport if the method is existing on any other instrument, so that method transfer also we do support. And further to that, we also support for method validation as per the user requirements.
So this question is for Madaswamy, since you mentioned about peak intelligence. So one of the questions from the user is that an optional software, or what are the software, like you mentioned about lab solutions inside peak intelligence and lab solutions. So what are the softwares that is required to do a honey analysis?
No, only lab solution inside software only for data processing. The peak intelligence is not a software, it's one of the function which was included in the lab solution inside software as a optional. So recently introduced by the Shimadzu, and it is working very fine when we done the study with internally, and we observed that this function is giving, and it is giving very good peak integration along with the combination with artificial intelligence. For example, high baseline with the low response compound, it will integrate automatically without any challenges. Just we will choose this function through this lab solution software, then it will integrate automatically. There was no different software, only one software, lab solution software. This is a different function, we have to add it in this software. That's all.
Yeah, thanks, MS. So this question is to Abhishek. Since you mentioned there are different models, because as a vendor, when you want to buy a new mass spectrometer, there are like many options that are available, either from the competitors or from Shimadzu. So what are the things that they need to consider while buying the systems? Is it the cost? Is it the flexibility? Or is it the sensitivity of the system? Or is it the future upgradability of the system? So what are the things that you should consider while buying?
Yeah, so this is a very interesting question, because as you mentioned, all of these points we have to consider. But ultimately, basically, what a user needs, I would suggest you should go with his requirement. It is not always recommended or required to go with the highest sensitivity model available, because a highest sensitive system will also have its own challenges. So in terms of handling, because it will be a very high sensitive instrument. So depending on the need, whether they are serving Indian domestic customers, or if it is for export regulations. So depending on your specific requirement, you can select a particular model which will suit your requirement.
Yeah, thanks, Abhishek. So this question is to Debijit. So one of the queries was, do you provide library along with the systems for identification?
Yes, absolutely. NIST library for GCMS is a very, very common universal library. As of now, NIST 2020 is the latest library which has launched. We provide GCMS with the library. The NIST library is the most common library what we provide. But of course, if customers demand for any other library which is suitable for their application, for example, some other libraries are available, Willy or pesticide library separately, or flavor and fragrance, all these libraries are also available with us.
Thanks, DJ. So there's one question from Alok Madeshia. So he asks, what is the role of acetonitrile and ethyl acetate in sample preparation? Probably Dr. MS can answer this one.
Whether you may use any organic solvent, whether it's acetonitrile or ethyl acetate, there was no difference. What we will do, only acetonitrile will extract the particular contaminants from the matrix. The purpose of organic solvent used to extract the contaminant solvent. Further, we will evaporate it through nitrogen generator. Then we will dilute with based on the instrument technique. For example, if you are using GCMSMS, we have to dilute with ethyl acetate. If you are using LCMSMS, we have to dilute with the acetonitrile or methanol or whatever the mobile base which was used for your particular analysis. That's all. Thank you, sir.
Thank you. And there's one question from Mallikarjun. So do you have workshops to learn these techniques hands-on for students? Probably Dr. Sagar can answer this one.
Actually, we have an institute called as Indian Institute of Chromatography and Mass Spectrometry. So under which we used to conduct a lot of training and workshop for students and industry employees, depending on the requirements. And we had some courses like 2 days or 3 days workshops as well. So with that, I think we can do that.
And Kisan Dasari, probably he must be using a old LCMS 8030. So one of the questions probably for Abhishek, the older models like 8030 and 8040, are these also upgradable to 8045, 50 and 60, or what are the challenges?
So see, since 8045, the instruments are designed on a different technology platform. So 8030 is upgradable to 8040, which is very much possible. But from 8045, there's a different technology platform. So from 8045, these are a different range of instrument. So 45 is upgradable to 50 and then 60. 30 is only possible up to 40, 30 to 40.
Yeah, I think that answers this question. So if you have a 8030, it can be only upgraded to 8040. If you are buying 8045 now, it can be upgraded to 8050 or to a 8060 on a later stage. Okay. Yeah. So another question is from Amitava: whether standard mix or single standard should be used. Please clarify the use of standard with respect to expiry date, especially after opening of standards. Dr. MS or Dr. Sagar?
Yes, yes, of course, we can use standard for any analysis because we are using analytical instrument. The instrument is quantification based on the external standard. Of course, when we are purchasing the standard, we come to understand the expiry date of the particular primary standard. Primary standard means what we have received from the vendor, whether it may be the liquid or solid. Then based on that expiry date, we have to prepare the working solution. It means stock solution first, then followed by the working solution. Stock solution is 1000 ppm. We may kept at 1 year. 100 ppm we may stored at 6 months. 10 ppm we may stored at 3 months. 1 ppm comes under 1 month only. Remaining working standard, you may prepare regularly for your analysis for the pesticides or antibiotics. So always till 1 ppm you have some expiry duration. After that working standard, you have to prepare daily and verify the standard and analyze the sample with external calibration only. It means working standard calibration only. Thank you, sir.
Thanks. So there's another question from Anuj Kumar. Kindly tell me how many antibiotics can be detected in a single run and what is the sample capacity per run and duration?
As per my experience with our Shimadzu LCM SMS, we have developed 126 antibiotics in a single run. Also, Shimadzu having the method package of these veterinary drugs. You cannot optimize the method, just we will pick the MRM transition which is available from the database. Same should be used for the sample analysis after verification of this method. In a single run, you can identify 126 antibiotics. Also, you can load multiple samples in your HPLC system. For example, N series with LCM SMS, we may load 162 samples in the one batch submissions. Depends upon the sample flow at your laboratory, you can load it maximum 162 samples. In these 162 samples, you have to include along with the calibration and QA/QC sample also. So decide the capacity is 162 is available in our front end. You may decide how many linear points, how many QC samples you have to include it. So one batch you can run almost more than 100 samples for this kind of antibiotics in a single run. Thank you, sir.
Yeah, thanks, MS. So I also have a query. For example, now there are LOQs which have been specified by FSSI and EC and this thing. So you develop a method now, and tomorrow, if the regulations goes much lower, the LOQs goes much lower. So do you have to again develop a method again, or do you suggest to develop a method with a higher sensitivity right now so that the method can be robust, and even if the sensitivity goes low or the FSSI or the EC says you have to go much lower, so the same methods can be used for the future?
Yes, sir. Earlier, the testing laboratory just focus the LCMSMS, but nowadays it is not like that, sir. The problem of the changing the regulation and the lowest level of the MRL value and as well as the LOQ value, the testing laboratory should be focused high sensitivity instrument hereafter. The high sensitivity instrument will give the better productivity. Also, you can identify the analysis very quickly with the lowest LOQ value. Whatever the method developed in the high resolution or high sensitivity instrument, you should not change more than 5 to 10 years. As per the regulation, if you want to change the method validation or you want to revalidate again, you can do it. Otherwise, the method will work out till end of this instrument working fine.
Thanks.
Thank you, sir.
So when it comes to GCMS, so what are the models that you specify, DJ? So is there any particular model or is there any difference between a GCMS and a GCMS-MS?
When it comes to GCMS, GCMS is a single quadruple, and then GCMSMS is basically known as triple quadruple instrument. GCMSMS for pesticide residue analysis, we highly recommend, because simple reason, for the closely associated peaks, you can resolve further, and then it happens in case of pesticide very much. We have suggested here what we presented actually is GCMS 8040 NX. That model is sufficient, having very, very high sensitivity, polarity switching, and then everything is very high standard, set at very high standard, which is sufficient for pesticide residue analysis or any kind of honey analysis application.
Thanks, DJ. And I see a few of them are raising their hands. If you have any queries, I request you to please type in your questions at the Q&A box so that we can post it to the experts and get their answers. So we have got another couple of questions. One, Mr. Anuj Kumar asks, sugar profiling can be done by LCMS. So why do we have to go for an HPLC?
So actually, sugar, as like Mr. Anuj has rightly said, sugar profiling can be done by LCMS also. However, sugar is considered to be an adulterant in honey, and whenever there is adulteration, it will be in high amount, like looking at the commercial aspects of it. So when the concentration is in percentage level or very high concentration, HPLC will suffice the purpose. And in any laboratory which is just to start the operations, so and they are having only HPLC in their lab, so they can do analysis using HPLC. Or a laboratory if is having LCMS instrument or they want to invest on LCMS, then obviously it can be done on LCMS also. Okay.
So even if they buy an HPLC, the same HPLC can be used to upgrade to an LCMS on a later stage.
Yeah, LC is a front end, so it can be connected to mass spectrometer at later stage. That is possible.
So there's another question. I think this is the final question from Prashant Tomar. So what is the alternative of amino columns for sugar analysis? Yeah, I was also wanted to ask this question since Madaswamy, since you mentioned there's a lot of columns from Shimadzu. So is there any specific columns that they need to buy, or do you need to like buy all the complete column spectrum?
No, for this sugar analysis, we are using Sodex's Helix Pack VG50. This column is giving very good response and very good separation. Also, the RT also very good. It means the elution of the sugars after 2 minutes. So our analysis, we have did this analysis by using this column. The fructose is eluted 3.5 minutes and glucose is 4.6 minutes. So this kind of the separations is very important. Also, the sensitivity also very important for this kind of the column. Is good for your sugar profile analysis.
Yeah, thank you so much.
Would like to add to this point. There are certain amide columns also available apart from amino columns. Amide columns are also there, and when we are using ELST particularly instead of RA detector, there is possibility of using gradient chromatography. In such cases, amide columns are also a suitable option where you can do separation of sugars using gradient chromatography.
Yeah.
Thank you.
So I think we have come to the end of the question and answer session. So if anybody has got any inputs to be given to our viewers, Abhishek or DJ, Dr. Madaswamy, if you have anything, you can give it now, or else we can go back to DJ. If his network is fine, we can continue with the presentation. Yeah, so we'll now go back to DJ. I hope your network is fine now and we can continue with the presentation.
Thanks, Mr. Satish. Sorry for the interruption between because of low internet bandwidth. I will try to quickly finish up. In fact, I was explaining about the turbomolecular pump, its importance for better vacuum throughout the chamber, right from ion source to the quadruple and to the rejector. Shimadzu uses a dual inlet differential turbomolecular pump design, which ensures that one part of it is kept just below the ion source chamber and the other part is kept below the quadruple chamber, which ensures that throughout the chamber you get a very uniform vacuum. So it ensures the maximum number of ions reaches to the detector, ensuring higher signal to noise ratio, which is nothing but your sensitivity. And here you can see, you can also use hydrogen as carrier gas in case of our front-end GC, the Nexus 2030. So hydrogen, helium, nitrogen, all three gases are very much possible to be used as carrier gas. Also, there is a function called twin line MS, twin line GC system. Here, you can attach two columns with the injector port, and it will be connected to two detectors. Here, either FID or MS, FID, MS you can simultaneously do. So it's called twin line system. At a time, you can inject to both the columns, and you can have the functionality of using the two systems at a time. This slide explains about the automation facilities of Shimadzu system. All four technologies are explained before. The active time management gives you a perfect time sequence of available works to be done. That is the maintenance, the tuning, the analysis, everything. It will automatically guide you through the times remaining for that. Smart EI/CI source, a single ion source, without breaking the vacuum, you can make the transition from EI to CI or PCI to EI automatically. Smart MRM database. Shimadzu has a smart MRM database which ensures that quick method development is possible, and then you can run through without any trouble. And ClickTech technology ensures that the column replacement or the injector port cleaning is very, very user friendly without the need of a tool. Smart operation. Shimadzu has smart MRM database, optimization tool, and lab solution insight. Here we can see that 530 pesticides MRM transitions it comes with the pesticide database version 2. It has two parts: compound information and transition information. All are automatically, the user is very much capable of. If he is not a very new novice user, then also it's very useful to use this smart MRM database. Lab Solution Insight software, as explained by Abhishek already for the LCMS. The same software here also can be used as a beautiful post-run analysis tool, wherein you can have the color flagging option in different colors, red or yellow. The yellows can be the threshold level which needs to be further checked for the yellow queue, and red means the red flag will automatically come which are not passed. So this automates the entire post-run analysis, which is a hectic for 2 days or 3 days job, or maybe many man hours are required. Here it is entirely automated. In just 1 hour, you can report everything. So for food applications, it is a very appreciated software platform by customers. 212 pesticides have been analyzed. This is the LOQ criteria. You can see the linearity curves, LOQ, specificity, and ultimately, out of 124 pesticides, all showed LOQ less than UMRL level. This is about Shimadzu MS platforms, whatever instruments are available today, different GCMS models. There are two single quadruple models, 2010 SE and 2020 NX. 8040 NX is the model what we suggest for the triple quadruple GCMS MS. And then 8050 MS is also another model available. Sensitivity. The two LCMS single quad models, the 2020 and very recently launched 2050 model. It's a very small and compact size machine with high capability. An LCMS platform. These are all the models available: 8040, 8045, 8050, and 8060 NX. This is all about the GCMS part. You can type your questions, then we can answer later on. Then I will just briefly explain about our HPLC portfolio. There are a few models: Shimadzu's P series, I series, N series, and we have compliant workflow for outstanding performance. When it comes to HPLC technology, basically in the market, you will see that there are three types of HPLC requirement from the market or the clients or the basic analytical industries. One is the general requirement of 3 to 5 micron column particle size, wherein we suggest the standard P series model. It's all around HPLC system, which is modular in design. Next is the mid-level UHPLC, we can say. It is UHPLC ready system. We have an advanced I series system, which caters to the requirement of 2.5 micron particle size columns. Next is sub 2 micron columns, for which we address with our N series UHPLC system. All three are very much popular in the industry. Starting with P series, it's a modular system. You can see all pumps, detectors, everything is a box, column oven, auto sampler. So it's a system which is proven performance with productivity. Over the years, many systems have been sold to all the industries. This system has the capability of automated system preparation. It's a very unique feature. What we call is, for example, in the morning in the lab, chemist comes and there is a sequence of activities what the chemist needs to do, right from startup to system checkup, the purging, the flow lines, column equilibrium, checking the baseline, system suitability checks, then your sample analysis can be started. Now here, all these steps which are manual earlier have been automated to one button called ASP. In the software, you click ASP and then you can start working in other activities like the documentation or any other work. You can stand in front of the machine. After a few minutes, you can come and you can see the green light glowing as ready. That means all these steps are automatically done. So directly productivity increase. This is the pump LC20AD. In fact, this pump uses 10 nanometer step size and absolutely lower pulsation. That means it's a pulsation-free pump, we can say. This is a very unique technology adopted by Shimadzu for all the absorbance detectors, be it UV or PDA. The temperature control flows the entire house temperature control. So whatever the ambient fluctuations are happening, there is no effect in the baseline stability. On the left-hand side, you can see that the room temperature is fluctuating, the black color detail line. According to the detector cell temperature also fluctuates, the red color line, and the baseline is also not stable. On the right-hand side, you can see that on the contrary that even if the cell room temperature is fluctuating, your detector cell temperature is fixed at a certain temperature, and in the process, the baseline is also very stable right from the beginning. This is a unique feature by Shimadzu. This system works as a perfect partner with the MS. That means you can integrate an MS. It can be single quadruple or triple quadruple. This is about the RI detector. This is our fluorescence detector, which is having the highest sensitivity in the industry. 2000:1 is the sensitivity what we can achieve with our fluorescence detector. And coupled with it, we can use the Pickering Labs photochemical reactor for the mycotoxins or the aflatoxin analysis. This is a beautiful combination, the fluorescence with the UV post-column reactor, which replaces the conventional Cobra cell. In fact, it is much better. Cobra cell is an old technology. which can be replaced with this technology, and then it gives absolutely fantastic result, higher sensitivity, much higher sensitivity. And this is AOSC accepted methodology. RI detector. Shimadzu is having a 4-partition cell design RID, which is patented in nature again. Same RID can be used for analytical, semi-preparative, or preparative. That means it can take higher flow rate up to 150 ml per minute, which no other RID is capable of in the industry. ELSD is also done by Dr. Madaswamy. ELSD is a technology which is very, very high sensitive technology. 10 times higher sensitivity it gives for the UV absorbance. It doesn't matter whether the compound is chromophoric or non-chromophoric. ELSD analyzes everything and gives a very high sensitive result. So our ELSD is the standard model what we promote for your applications. This will be a very good model, and it comes with different types of nebulizers. It can be analytical nebulizer, or it can be ultra fast HPLC capable. So we have all. Next HPLC model is called I-series, advanced I-series model. This is the picture, and it comes with analytical intelligence. The first time ever introduced in any analytical instrumentation by Shimadzu. You know, the living technology, the voice commands, many things that automates the entire processes. Like human brain, it can work. So it adapts the intelligence of analytical in the analytical industries, the analytical workflows. It acquires high quality reproducible data and independent of operator skill level. That is very, very important. Your operator may be a novice user, doesn't matter. The system will take care of it automatically, and it is going to apply its own intelligence. This is the complete 24 hours automated workflow, like auto start to shut down, everything is automated. So it's a 360-degree automated system preparation which works with auto judgment capability, minimum human intervention, and also you can monitor your system from remote locations with your smart devices all the time. It may be a smart device, maybe iPhone, it may be any kind of smartphone. You can install the lab solution direct in app format, and you can monitor the system from a remote location also. This high is the technology. It also uses a work from anywhere kit, WFA kit, what we call is, which is a gravimetric sensor which is being used for the solvent bottles rack. That means if the solvent runs out below a threshold level, it will automatically shoot a message or alert to the user's device that mobile phase is not sufficient. You need to add some more mobile phase. In post-COVID or during COVID time, it is very, very important that you are working from home or you are working from remote locations. You need not stand in front of the machine all the time. This really automates everything and eases the user's life. This is the auto sampler front axis, and it has dual temperature, individually temperature control 2 racks, which ensures that even if your first rack is having under analysis, the second rack you can insert your samples and it will not hamper. So you can directly increase your productivity of the lab. It ensures the carryover is absolute minimum for the auto sampler with very high throughput. Coming to the N series, our UHPLC model benchmark, and it has 5 series. Like it is also analytical intelligence enabled system with the mobile phase monitoring option, MPM kit. Everything is available with some innovative designs. This system is based on 42 patented features of Shimadzu, which ensured that Shimadzu backed the Red Dot Award Design Award. So setting up the instrument, running and post-run, everything is automated here. Setting the mobile phase, auto purging, equilibrium, start the analysis, data processing, shutdown, and then it also has a beautiful feature called flow pilot that will be explained. Generally, what happens? The columns are treated as consumables, but definitely many columns are there which are expensive in nature. and making the condition for the longer time is very important. What happens generally? Left side, you can see there are two diagrams, left and right. Left side, without the flow pilot, the general phenomena what happens till the column oven comes to a stable temperature. Till that time, your flow is increased. Maybe it has been set up to 1 ml. So your flow increases up to 1 ml and temperature is gradually increased in the column oven. That means it automatically inserts higher pressure in the column. So chances of column getting damaged is very, very high. What has been done by Shimadzu R&D is, in order to prevent the columns, the flow is set half of the set desired flow rate. That means if 1 ml is desired, you are setting 0.5 ml initially. The system sets automatically the set temperature. This ensures that it will not give pressure to the column. Column life is much higher. It is protected for a long time. And we have method scouting kit. So multiple columns and solvent can be automatically switched over to come to a method development solution with a software called method scouting software, which makes the life much easier. Many man hours can be saved for automatically developing methods. This is running 6.0 seconds injection cycle. You can imagine how fast the system is, how many samples you can do in a day. With a rack changer, which can provide 17,000 samples which can be analyzed, highest throughput. When it comes carryover, the system gives absolutely minimum carryover of 0.0003%, which is nothing is detected. So you are assured about the quality of the system with respect to carryover and faster injection. This system is having automated sample pre-treatment facility with all this reagent addition, co-injection facilities, auto dilution. Thank you for your attention.
Yeah, thank you, Devjit. So we have come to the end of today's webinar. We still have one query. I think we can take that question. So it was answer. Yeah. So Mr. Anuj Kumar asks, there are 126 antibiotics in a single run and totally 162 sample capacity. But what about sample runtime per sample for antibiotics?
Yes, sir. Very simple, this method actually. 126 antibiotics in a single run. If you are using ODS column, the runtime will be only 16.5 minutes. Suppose if you are going to use 5-phenyl column, the runtime is less than 15 minutes. So the column will be chosen based on your matrix. Decide yourself, but the runtime should be always less than 20 minutes for your analysis for single sample analysis. That's all.
Okay. Thank you so much. I don't think there's any other questions. Thank you, panelists. So we have come to the end of today's webinar. Thank you so much for joining our webinar and your overwhelming response. So we have received your request for the application booklet and will ensure to courier in the next few days. Please do follow our LinkedIn page so that you can get all the updates about our upcoming webinar. So once again, from Team Spinco, we thank you for attending our webinar and wish you a very wonderful weekend. Thank you so much.